Hello everyone, this is 𝚂𝚎𝚑𝚘𝚌𝚑, BSc Zoology, 𝚂𝚎 College. Today, my seminar topic is class Trematoda, example Fasciola hepatica. Flatworms are highly diverse in their mode of life, functional biology, and evolutionary status. Class Trematoda comes under the phylum Platyhelminthes. What is Trematoda? Trematoda is a group of parasitic flatworms commonly called flukes. The distinctive features of the group are the following: flat, leaf-like and unsegmented adult body without ciliary covering. The body is covered by a non-cellular living layer called tegument, which is a dense mass of cytoplasm. Adhesive organs include oral and ventral suckers and in some cases hooks also. Epidermis does not exist as a discrete cellular layer in the adult. Reproduction is exclusively sexual. Most members are hermaphrodites. Excretory and osmoregulatory organs are protonephridia with flame cells. The life cycle is simple or complex. It's a complex life cycle involves several larval stages and one or more intermediate hosts. The most common example for class Trematoda is Fasciola hepatica. Fasciola hepatica, popularly known as liver fluke or sheep liver fluke, because mostly it is seen in sheep. It is a parasitic flatworm of the class Trematoda, which comes under phylum Platyhelminthes. It is a pathogenic endoparasite Trematoda inhabiting liver and bile duct of mammals including humans. The disease caused by flukes called fasciolitis, also known as fasciolosis. Fasciola is a digenetic parasite because its life cycle is completed in two hosts. Definite hosts include sheep, cattle, mammals, accidentally humans. Intermediate host is freshwater snail. The infected animals become anemic and lose significant amounts of weight. Morphology of Fasciola hepatica. Adult liver fluke has a soft dorsoventrally flattened and conical body. At its broad anterior end is a conical projection called oral cone or apical cone or head lobe. Mouth is situated at the tip of this cone, encircled by an oral sucker, and ventrally, a little behind the oral cone, is a posterior or ventral sucker often called acetabulum. It serves as an adhesive organ for attachment to the host. Friend of acetabulum is a common gonopore. At the extreme posterior of the body is the excretory pore opening. In addition to these apertures, a temporary opening of Lawrence canal appears during the breeding season on the dorsal surface just anterior to the middle of the body. Life cycle of Fasciola. In the beginning, we see that life cycle of Fasciola is complex one because it includes two intermediate hosts. The life cycle is highly complex and it includes or it involves 5 larval stages, namely Miracidium, Sporocyst, Radia, Cercaria, and Metacercaria. The fertilization is internal. From a simple flow chart, we can understand the life cycle of Fasciola. The fertilized eggs are stored in the uterus for some time. During this, each egg gets coated with yolk and enclosed within a shell. Now, embryonic development begins and shelled eggs transform to shelled embryos. Then they are discharged to the bile duct of the host. From there, they reach the intestine of the host and finally pass to the outside along with host fecal matters. Under favorable conditions of light, moisture, temperature, and oxygen in the soil, each embryo develops to a ciliated larva called miracidium. We are going to see the 5 larval stages of Fasciola hepatica in detail. First one is miracidium. Miracidium is a minute, free-swimming ciliated larva. Its body is composed of outer epidermal, middle muscular, and inner mesenchymal layers. Epidermal cells are ciliated, 18 to 21 in numbers, and arranged in the 5 circular bands. At the anterior end, miracidium has a conical and protrusible projection called apical papilla, or it's also known as boring papilla, head papilla, or terebratorium. Lying inside this papilla and opening at its tip is a saccular apical gland, a pharyngeal gland, and a penetration gland, or also called cephalic gland or lytic gland, are also seen on each side of a apical gland. The secretion of these glands serves as histolysins to dissolve the snail's tissues for penetration. Near the base of apical papilla, a nerve ganglion or cerebral ganglion and 2 pigmented eye spots are also present. Inside the miracidium, there is a pair of protonephridia. Seen in the posterior half, there are several large undifferentiated embryonic cells called germ cells or propagatory cells. Some of them undergo repeated division and form small clusters called germ balls, which give rise to new larvae at a larger stage. Miracidium can undergo further development only in the intermediate host of Fasciola hepatica is the pond snail Limnia truncatula. The larva has only a very short free life. Within this period, it has to gain access to the snail, otherwise it would die. It bores through the mantle and the pulmonary sac of the snail with the help of apical papilla and tissue-dissolving secretion. Finally, it occupies the roof of the pulmonary chamber. It undergo for the further development the roof of the pulmonary chamber. It casts off its ciliated epidermis, apical papilla, glandular apparatus, cerebral ganglion, and eye spot also disappears. It penetrates to the internal tissue and transforms to next larva sporocyst. Second larva stage, sporocyst. Sporocyst is an elongated and saccular larva. Its body wall consists of outer tegumen, middle muscular layer, and inner mesenchymal layer. Internally, sporocyst has several germ cells and germ balls, those of miracidium. Germ cells undergo repeated division and form further germ balls. On the other hand, germ ball develop as embryo. Some of them directly develop to third type of larvae, radia. This phenomena by which several embryos are formed from a single zygote called polyembryony. Fully formed radia comes out of the sporocyst through a temporary opening called birth pore. Soon they migrate to liver, hepatopancreas of the snake. Third larval stage, radia. Radia is an elongated and cylindrical larva. Its body wall is composed of outer tegumen, middle muscular layer, and inner mesenchymal layer. Near its anterior end is a muscular band known as collar, and near its posterior end is a pair of ventrolateral processes called lappets. Collar serves for a locomotion and anchorage. Close to the collar is an opening called birth pore. Radia has a short alimentary canal, and a pair of protonephridia has a small apical mouth, suctorial pharynx, short esophagus, and circular intestine. Surrounding the pharynx and opening to it are numerous pharyngeal or salivary glands. Radia has numerous germ cells and germ balls. They escape from parent radia through birth pore. In winter season, the germ balls of primary and secondary radia develops to the next larva, namely Cercaria. Fourth larva stage, Cercaria. Cercaria is an oval, flat and tail larva. Body is covered by a spiny tegumen. It has two suckers, namely oral and ventral suckers. The whole interior of the larva is filled with parenchyma. Laterally, just beneath the tegumen are numerous cystogenous glands. Cercaria has a well-differentiated alimentary canal, a pair of protonephridia and numerous flames. Alimentary consists mouth, suctorial pharynx, long esophagus and a forked intestine. Two protonephridia terminally join an excretory bladder lead from excretory duct opens out by an excretory pore. Fully formed cercaria escape from radia, reach pulmonary sac of snail and finally escape to the outside. The final larval stage, metacercaria. Cercaria has only a short free life. It swims about in water for a few hours, then settles down and finally attaches itself to a water weed with its ventral sucker. Its tail is lost. Its cystogenous gland secrete 4-layered cyst around it. This uncysted cercaria is called metacercaria. Metacercaria is infective. Its further development is possible only in sheep. Sheep often swallows large number of metacercaria while it feeds on the plants to which metacercaria attached. In intestine of host, excision occurs under condition of high temperature, high carbon dioxide tension, the presence of bile, etc. The excisted metacercaria penetrates the gut wall of sheep and pass into peritoneal cavity. They migrates to liver and from there it gets matured to the udder fluke. In about 3 to 7 weeks after infection, they invite to the bile duct. So that all about the class Trematoda and its example Fasciola hepatica, small fowl G and its life cycle. So thank you all.